Good evening, students. And this is the continuation of the second experiment of your bioinformatics lab. In previous experiment, you have done PDB structure viewing and analysis by using Chimera, right? And this experiment, you are going to classify a protein, your protein of interest, and you are going to search that and which classification is comes under, and what are the active sites, and how many amino acids are there, and what the alignment with the other species. So what is the domain, how many domains are in that. So everything you can get by using the structural information. And the online resources we are going to utilize are SCOP, structural classification of protein, and a CATH, then ProSite, right? This thing. So the applications will be, you can get a basics idea about the classification and the functional classification of the protein, and you can get a G also, gene ontology diversity, right? This is classification of that protein, enzyme classification, species diversity, everything you can get. And also structural neighborhood also, if you want, you can find the topology of the protein and how many proteins are similar with that also we can find. And here, like a cat scope also, another server, you can find the family and the protein summary, proteins, domains, and architecture. So the same thing you can get it from UniProt also, but here you can get the amalgamation of informations from various sites. Domains, you can explore the details of domains which are there in that. So after that, you are going to use ProSite, that is protein site, nothing but. So in this ProSite, you can give uh, for a protein sequence or a name in the search box for searching the motifs of particular protein. So you can give the sequence of that FASTA format sequence, and you can observe what are the results, how many proteins are aligned with your protein of interest, and the what are the scores and active sites present. Especially we can use the site to find the active site of our protein, and basically you can also find the length of the sequence or count of the amino acids present in that. Okay, so shall we go to the experiment now? And this is the next experiment for you. We will discuss in detail in the next class. Okay, and first of all, we have to go for cat, right? Okay, so this is, you can just type cat and that you can find the protein structure classification database by at UCL. There is a University of California, I think, right? So in each and every universities in foreign countries, they have very good bioinformatics resources and servers and their own lab servers, like modeling, structure prediction, and ADME toxicity prediction. They have their own tool, and it also, they also make it as public. Okay, anyway, this is the thing. So you can find 3D structure, protein evaluation, and a protein function, console site, and you can download the data about. Anyway, so I will just put it here, a glucokinase, and these are the previously I have searched for you, I think. I don't know what kind of protein it is. Anyway, I will give it. That is a PDB code, right? Don't worry. The four-letter codes are PDB codes. If you enter the PDB code, means directly fetch the information from the PDB sources, protein data bank. So the matching is fine. Cat families. Instead of giving PDB ID here, you can give your protein name also by text, right? For example, previously I have used hemoglobin. I can just type the hemoglobin, it will show the results. Okay. So we will see that probably this is a globin, maybe this is a PDB ID of the hemoglobin, and defaultly it was there. Okay, so matching cath superfamilies, how many matches are there? So you can click here and you can see that globins PDB code is 1A3N is the chain A domain, and PDB code. These two types of structures available in the matching PDB. So in the results you have to mention globins. Okay, so if you want to see that, you can just click in this. You can find unique gene annotations, geotones, gene ontology and mutations. You see that response hypoxia. You can just know that, and you can find what are the terms related to the globin. And this is easy. You can find cyclase enzyme classification, right? Nitric oxide, mostly it is also nitric oxide and dioxygenase. And the species, you can also find others in this thing, bacteria, invertebrates, and here plants and fungi, primates, vertebrates. So we will come to the vertebrates. So these are the species diversity. If you want to take a snapshot, means you just click here and move the cursor and take a snapshot. Otherwise, these pictures will not give any information in use. Okay. So otherwise, you can just put. You can edit this. Copy and paste the pictures in your PPT. In PPT, you can edit. Like means you can create an arrow mark and put Homo sapiens in this. Otherwise, which is a big oxygen transport. Like that, you can show by using arrow marks in the PPT. Then make the PPT as a JPEG. You can paste as a result, right? And these are the functional families of that globin. If you want to deep research in this protein classification and homology, means you can go for that. We can go back and just give the click on that. Okay, so you can find everything. Structure classification 1 and 2, how many proteins are there, and this is about the phylogenetic trees like phylogenetics. See isoforms, how many structures are available. So everything you can get it from here. And sequence similarity, we are not going to analyze this for this now in this experiment, so you can omit this. Structure domains, there are 2891 domain clusters. More than 95% sequence IDs are available in the public databases, which are 200 IDs are available. Domain clusters are 52. Unique PDB IDs, see that nearly 1224 structures, PDB structures are available in the PDB itself for that. Okay, function. Unique QC is three kind of functions that will go. Unique GO 44. These three unique. What is that? Unique EC, right? And this is EC. And see that digonalate cyclase, others, and nitric oxygenase. And here oxidoreductases. These are the functions of your protein which is selected for the analysis, right? Like that, you should show your own protein of interest, function, and diversity. Okay, this is about a cath. You can find the families, superfamilies, right? Classification of domains, and see that mainly alpha, orthogonal bundle, right? Mainly alpha we need, and this is what. This is very important. You can put it as a results in your protocol, which means in your observation notebook, and see that you just explain this. family, super family, more than 35% means this color, and 60% is the dark gray is 100%. Okay, you can click here and find the clusters and domains of your protein relation. Okay, so you can just see how many domains are there, how many see that which are mostly related to more than 60 is notable one, right? More than 60 you can write, which means you have to find out how many are there in the structure in this pictorial uh, explanation. You can find that how many is R falls under more than 60 and more than 95 and more or less equal to 100 percentage. Okay, so you can just what is the cluster is there, how many domains are there, and how many domains are there. So in this just you can put the highest one. For example, you can use this clusters. 552 domains are there in the cluster. The and here the cluster classification is there. If you put the same cluster classification in this cath, it will give the exact information about your other cluster. In which cluster you want to work. Sometimes we may work on the protein family, how they are comparatively. In the research we may select to how the proteins are working in families, which means the same family of proteins are with some mutations and wild type, how they are working and what is the rate of the reaction of that protein or how many factors are affecting the enzyme. For those studies, this will be helpful. Okay, so this is about cath. And this thing also you got to put as a screenshot so that you can find diagonal cycles. We already found it with the EC, right? Enzyme classification pictorial definition. And that, okay, here you can find that. Okay, how many structures available are not hemoglobin subunit? What are the alignment diversity? 0 to 100. So you have to take the snapshot from 61.4, right? And this is 75 is there, again 90 is there. So can we see, you can put here and you can classify them, so you can get the highest one. You can just copy, or you cannot copy, you can take a snapshot, use the snipping tool to make this snapshot. You can take it from 61.4, what is that, neuroglobin. From that you can select up to first one, top hit, and that is 97.5. You can just paste it. So by this way you can easily find out how many proteins are aligned with your protein, what are the, is there structural information available in PDB. These are the extra crystallography or NMR solution structures, right? So what is the sequence length of your protein of interest? So this is how you can project your new result. And then a structural neighborhood, some here there will be a graph, so we need not to put how they are relative. So unless otherwise, if you are doing a topology analysis, means you can go for this. Okay, so you best of me this also, no need to do in this experiment, and we will see if we are working in a protein depth research. Okay, so this is about cath, and the same thing you can do with SCOP, right? So now you can find you can give the protein ID, so it's given, right? So you can the protein you can give PDB ID or text the name of the protein. Then you can find the superfamilies, go and find the geo diversity, EC diversity, specific, then classification domains there, that is most important. Functional families are in the left hand panel. You can just click it and you can paste the result, right? So domain in chain, you can, if you go to the domains, you can find the domain in chain, how many chains are there in that. So likewise, you can repeat the SCOP, structural classification of protein, same thing. So which family it is belongs to, and PFAM is there. The olden days we are using, now it is integrated with the Interpro of EMBL. So domain, yeah, already you can export, but you can check the details with the SCOP also. Okay, so we will go to that SCOP, structural classification of proteins. And here you can just use enter free text SCOP ID PDB ID or UniProt ID, right? And here I will give the same thing. You can use go. So you can give your protein of interest for this practical purpose. Okay, so there are 8 represented results have come, and you can use any one of this. I don't think so. For you, you will get only one result, most probably. See that? And this is how the domains. See, domain in chain is there, right? Domain in chain and domains only. This is the entire chain, which are domains in the chain, and here is the domains only. PDBs are not given here. And the thing is, you have to select show ancestry. If you select here means you will get a detail of hierarchical detail. See that alpha class is this thing. Forget this. All alpha proteins. Fold globin like fold. Superfamily globin like family. Family globins. Domain is there. So if you click here, it will get you in the specific domains. Okay. So this is hemoglobin subunit alpha. Species is Homo sapiens. Just you can click that. Okay, this is how you can use this software, which means server. And externally, if you want, I already told you, I think DALI is one of the structural database. And this was the question, I think 3 years before. In that 2 marks, they have asked in an university what is DALI, right? So just read this. You are familiar with the PDB or SPPDB, UniProt, CATH, ECOD, nothing but the classification PDB. And this is KB means knowledge based. Okay, UniProt KB. Actually, we should say UniProt KB. Then DALI, top match, and fact. I am not working in this still. So PDB fold is there because we have uh, tools, servers, and software, standalone softwares, online softwares for everything. In that, we are using only the best, which means the scientists who are working more and more in the particular software means we are choosing to work on that. So no need to go everything in this. For example, there are so many folding to find a fold or make a fold softwares are available along with the PDB E fold, right? Okay, this is about scope. Then what is the next thing? So now you can show ancestor you shown and overview proteins domain architectures you can and see in this, right? So the domains in sequences, how many are there? So representatives will be here, and you can go for, suppose in this space you will find the PFAM actually. Okay, in this they didn't give, I think. Anyway, we will move to the next one, right? So PFAM should be here, is there now, and they should be in that scope ID they have given PFAM. Okay, we will check PFAM in that. individually. See, PFAM is now hosted by Interpro. Mm, Interpro example. I can just paste if I want to do it. So, for example, I will go. In this, you can search. They have modified everything. Okay, you can give the keyword. Otherwise, I will. If you give the keyword search means you can go for Go. Now, at the practical purpose and time being, I will give the example. What is this? Now I will give the Go. So that from this you can find the summary and domain architecture, everything. See that by sequence, by text. So search is going on, I think. See, these are the results. We have found 13 entries in that protein phosphate EAA. I have given as an example. I didn't give hemoglobin here. So you can give your own protein and you can find that, right? So we can go explore by sequence, and this I can give as a sequence, FASTA sequence to find, and here by domain architecture. Architecture must include, right? So here we will see in the next class about this in detail. Now PFAM, right? Now you do one thing. You just go and search the sequence by using, for example, hemoglobin. I will search and let us know. See, hemoglobin is there. Alpha, beta, theta, later HB subunits are there. We'll go on B dependent globular domain of extracellular hemoglobin linker. Just for example, I will click here. See that? You can find the databases and short name linker and description. You should copy and paste as a result whatever you got, and see that proteins, how many taxonomy you can find, how many proteins are there in that. See, in this, what you have to do is you need only reviewed, right? So you can find only the three, and this is matches of. Okay, so you just make a oh, all are alpha fold. You see, so the next experiment we are going to do in the alpha fold. Okay, you just copy and paste, or you take a snapshot and paste it. How many you can proteins results. We have only the proteins of tube worm. Protein has matched structures. We have how many PDB structures are there? See, nearly 4 structures available for that. And AlphaFold, see, 140 structures are deposited or created by the Google and EMBL. And this AlphaFold is made by the collaboration with Google. EMBL collaborated with Google to make an protein structures, right? You see, 140 protein structures are available. They have created and deposited in that. So these are the results you can find in this. See, information alpha fold model confidence. They have, if you select any model, and they have given the PDB 3D structure also. In the next experiment, we will see detail about AlphaFold, right? Nothing but we only 5 minute search in AlphaFold, but we should go for Chimera after AlphaFold search. Okay, thank you.